streaming on my YouTube. So my genetic counselors today and later on also, they will hear from you. And I have gone through your slide set. So this will be more for the background and a general. So in details about each syndrome, especially the Lynch syndrome and HBOC, that they will be covered in detail, much detail later.
So they know
So
all about that. Okay.
yeah, no, not only they know, there'll be a detailed session on that, so you can talk more about the general things and the background, and
Okay,
those
fine.
aspects you
Yep.
can just cover in general, especially the Lynch and HBOC, which we will cover, but other syndromes. So
Okay.
all yours, Shirley. Thank you very much for agreeing for this lecture. And I was telling them you have been coming to all our ICGC workshops, and we had such
Mm.
good time of learning and having fun as well.
Yep, it was very wonderful.
Yeah, thank you. Please, Dr. Shirley.
Okay, well, I went to the dentist yesterday and he told me that the first thing I should say is to congratulate you, the India, for beating Australia at the weekend in the cricket. So that was nice. Good for
Okay,
you. Very
thank you.
difficult. Mm.
Yeah,
And on their
yeah,
own soil. Good.
yeah.
So anyway, I was going to start with Mr. Mendel, who I think
So
you
before that,
are.
Shirley, if I may ask, you can just briefly, because now we have a formal session, introduce yourself.
Mhm.
And tell us how you went into genetics, because this is a recorded session. Go back,
Oh, I
please. Brief.
see. Well, as I was saying, I tried not to go into genetics because my father was quite a well-known geneticist. But I got a locum at Guy's Hospital when I was actually working in general practice. And I just found it so fascinating. I couldn't not do it really. And also with cancer genetics, I got involved with that because I just found out about Lynch syndrome and family cancer clinics being run by Joan Slack at the Royal Free. And my cousin, her mother was being looked after by my husband because she had her second cancer. She had colon cancer and uterine cancer. And he thought she looked rather like my cousin's wife. And he found out that she was, in fact, her mother. So I sort of stormed around and I thought perhaps my cousin's wife should go off and see somebody about this. And it turned out it was terrible. She got ovarian cancer and she died. And the reason was that her mother had Lynch syndrome. And that really sort of spurred me on that it was really important to do genetics of cancer genetics, because that was where you could actually make a big difference, perhaps in preventing people from dying of cancer if they had a susceptibility. So that was how I got into cancer genetics in particular. So, you asked for it.
Yeah.
Anyway, so shall I start with the Mendel?
Yes, please. Now you go as per whatever you want to
Yeah.
say.
Oh, and just to say, of course, thank you very much for asking me, because it's a great honor and a pleasure, and I love to be involved still with you guys, and hope we can continue our collaboration. So there was Gregor Mendel. They didn't really know much about genetics until he started fiddling around with his botany. He was a monk, but I think he spent quite a lot of time doing botany. And I'm sure you all know he started doing crosses between peas and flowers, which had different characteristics. And the characteristic one here was that he bred green beans with each other, and found that some of them were yellow. And when you did the studies, actually the first study is, whoops, this is better. He bred the smooth beans with the wriggly beans, and in the first generation they were all smooth. But in the second generation, he got a ratio of 3 smooth to 1 wriggly one, and that worked with quite a number of different characteristics. And so he was very clever in that he worked out that there was a law of segregation. Basically, there must be some factors that you would inherit, which gave rise to these characteristics in the phenotype. And there were various consequences to that idea. One is that dominant characteristics masked the effect of the recessive one. and that the genotype was very important, and this was a totally new concept in those days. And so basically, it led to the deterministic theory of genetics. And I mention that because there are a number of people now who are saying, oh, that's not right because there's a lot of genetics which isn't due to these strong genetic factors. But I don't agree. I think obviously it's a very important starting point because we know that there are quite a lot of genetic conditions which are caused by single gene defects. But to go back to this slide, of course, diseases can have hugely varying components. Some are definitely due to a single gene, but an awful lot of conditions are due to perhaps infections or all sorts of environmental things, and indeed other genes as well. So it's not at all straightforward. And when Mendel discovered this theory, when he in about 50 years before it actually got caught on, and it was revisited in about 1900. And also they did some of his work again and found that the answers weren't quite as clear-cut as he had said, so that it did cause some consternation. But they rediscovered Mendelism in about the 1900s. And also they used this kind of a theory to work out how evolution might work. But there is an awful lot of non-Mendelian inheritance about, and so I thought we'd talk quite a lot about that.
So, Shirley, if I may just ask, so
Mm,
use the word Mendelism. So, it's not very commonly people say that. So, could
mm.
you just elaborate? And when did you first hear about Mendelism? Did your father ever used to talk about Mendel? Or because he was in
Um,
population genetics, I suppose?
Not agree. No, not well. He'll come a bit later. You'll see a few things that he did.
Okay.
Um, I suppose. I mean, it was just Mendel's ideas. That's all.
Okay.
That were being
Okay.
appropriated.
Okay.
So anyway, so what do these genes do? Well, the Fleming and Strasberger described chromosomes in 1882, and they were found also by some other people in the 1860s to be derived equally from sperm and egg. So that was important because the sperm and egg gave you a haploid um, contribution to the diploid person or whatever it was. So that was a very important concept as well. So you had two haploid germ cells which went to make the mature cell that grew into the adult. And so the chromosomes split into two during reproduction. And obviously it took quite a long time to find that the genetic material was found to be DNA. But just at the beginning of all this, so in the 1940s and 30s, they were beginning to have a look at these chromosomes in little rows like this. And it was in, oh dear, this has been, here we go. Down syndrome was described by Langdon Down in 1866. But it was only in 1959 that it was found that it was due to having a trisomy of chromosome 21. So that was the first way of demonstrating this. I don't know why this slide's doing these funny things, sorry. Um, so then my dad did a study in culture. It was called the Colchester. It was a place where people who had mental deficiency, as they called it then, were incarcerated and looked after. And um, he obviously had a lot of Down syndrome children to have a look at. And he noticed that the age of the mothers was very much higher in most cases than usual, and that he thought maybe this had something to do with the etiology of Down syndrome. And he worked out that the father's age didn't seem to matter. And so 95% of people who got Down syndrome have trisomy 21, and this is caused by non-disjunction. And it is thought that in the female, the eggs start to divide actually before birth, and they just sort of hang around in this half disjuncted state and until the egg actually is ovulated. So clearly that's quite a damage-prone time, and that's probably why it's more likely that older mothers will have trisomies in their offspring. And in men, I think the sperm just sort of divide all the time and are produced all the time, so that that issue doesn't arise. But we also know that about 4% of children with Down syndrome actually have it because of Robertsonian translocation, which is therefore much more heritable. So the parents might be carriers of a balanced translocation. And so the recurrence risk for the parents of a child with Downs who's got a translocation is obviously a lot higher than with the free living trisomies. And then I remember my dad describing a few of the mothers of these children actually having some features of Down syndrome. They call it Mongolism in those days. Um, and so he wondered whether they might be mosaic for a trisomy 21. And so that they again would have a higher risk of having another child with Down syndrome if they had a significant mosaicism for that themselves. So then people got interested in the possibility of children have abnormalities being due to not just a whole trisomy, but a small piece of chromosome being missing or extra. And they developed this thing called FISH, which is fluorescent in situ hybridization. And in this particular case, it's for the whole of the chromosome. So you can see the chromosome 21 probe shows up as 3 copies in this interphase nucleus, whereas there are only 2 copies of chromosome 18, which is what you should find. And that was a nice way of showing this in the interphase. But also, they started looking at little portions of chromosomes so that you would get a probe, for instance, for a particular region. And then you would stick onto this probe some fluorescent antibodies, which you could then put into your chromosome mix. And in this is a chromosome spread, in fact, but it shows you chromosome 22 region, which is being lit up there. And you can see on the left, you should have, there are two probes being used there. And so you should get 2 copies of each probe in the normal one. But there's a child with a deletion in chromosome 22, where you can see that and 1 copy of the chromosome 22 marker is missing. So that was a good way of showing that this guy had a deletion in chromosome 22 in that particular region. But of course, people don't use that much nowadays because it's all down to looking at the DNA in much more detail. So hey ho, you find the discovery of the double helix in 1953 by Watson and Crick. And that was very nice because it showed you that DNA was a linear sequence of nucleotide bases. And that generated a complementary sequence of bases on the other strand, and it was held in this wonderful helix. And it allowed then for DNA sequence to be translated into RNA and then into proteins. So that allowed the idea of a gene to be developed in much more detail. These are not very clear, but this is a sort of picture that when we were getting of the structure of a gene in 1970s, where there were exons and introns and non-coding regions and so on. And this was all fairly standard, but it was terribly important to understand the way these genes worked. And so you could also discover how the transcription worked from DNA to RNA. So this was regulated by promoters, and then the RNA polymerase copied the DNA into RNA. And then the processing went on before you had to splice it so that the introns were removed and so on. So it's all really very clever. And I remember I was working on Duchenne dystrophy in the 1980s, and they found the gene when I was actually working in the department at the Hammersmith, where they looked after a lot of these children. And I remember we were very interested in the fact that you could get mutations in the same gene and you could get completely different severity of disease, which is Becker or Duchenne. And Becker is obviously much, much milder than Duchenne muscular dystrophy. And so we couldn't work out how you could get a big deletion in the gene and still get a mild form, the Becker, whereas you might get a tiny deletion and you get Duchenne dystrophy. And I remember sort of looking at these mutations to try and work out why this might be. And eventually it became obvious that in-frame and out-frame, in-frame deletions were much more likely to cause Becker because you could go, even if it was quite a big deletion, you could then go back into the reading frame without it having been destroyed. So you could get a reasonably normal copy of the protein. Whereas if it was out of frame deletion, then you destroy the gene and you get Duchenne dystrophy. So I remember that was very fascinating.
So, Shirley, before this Duchenne, the gene was known. What were you all thinking? Is it like a chromosomal disorder or a single? What was the thought
Before, no,
process?
I think
Yeah,
we thought it was a gene.
you
Yeah, I
thought it
think
was a
so.
gene because
Yeah.
chromosomes were already well studied and they must have been studied all the
Yeah, I
time.
think so. They were pretty major
Yeah.
disorders, but well,
Okay.
Duchenne is too, but anyway,
Yeah.
don't know.
Okay.
Anyway, a lot of work then went into the positional cloning and finding where these genes all were and sequencing them. And so there's a lot of work going on after the discovery of DNA. And then the Human Genome Project, which was a huge project to sequence the whole of the genome, which was completed in 2004. And I think there were two or three other groups which did that, but there was one in the UK. And so you've got 20,000 protein coding genes. But the very fascinating thing is that we've all got almost exactly the same number of genes as worms. And we have about the same ratio of non-coding and coding genes as worms do. So why are we so much more splendid and complicated than worms are? Well, the only difference you could show is that there are much more transcripts in humans than there are in worms. So presumably it's allowing these different genes to be turned on and off in different situations that is actually causing these complications in humans, as opposed to poor old worms who are very nice in themselves. So we'll come back to that because obviously gene regulation is terribly important. But if you go back to looking at the information that we got out of all this, you find that coding genes account for only 1.5% of genomic DNA. So there must be an awful lot of complicated non-coding DNA which actually regulate regulate gene expression. Because I mean, if you think about it, if your liver and your brain have the same DNA in them, but why isn't your brain like your liver? Well, it's because different genes have been turned on and off. And how do they know to do that? It must be very, very complicated set of regulations which go on. And I guess that's the crux of it, really. So then the next question was, everybody said, "Oh, the human genome has shown us what all the genes are, so we'll understand everything about common diseases, and it'll all be quite simple." And of course, it's not like that at all, because most common diseases are polygenic and multifactorial. And the genetic information that we can understand about only causes usually less than 30% of the causes. So you're thinking about asthma and heart disease and diabetes and all sorts of things. Well, most diabetes anyway. And so what is the missing heritability that we can get a handle on? Well, there are, of course, maybe rare variants that we don't know about or gene-gene interaction, but there's an awful lot of environmental influences and imprinting that is important in how it all works. Now, just to go back just a moment, though, to think more about diseases and how much genes affect them, obviously there's very important studies that were done by Tim Spector and people at St. Thomas's and many people before that had shown that a lot of studies of monozygotic versus dizygotic twins were being done, and you ideally you would find that single gene disorders would be 100% concordant in monozygotic twins, whereas dizygotic you would get the same sort of concordance as you would in siblings. And if it was an environmental thing, they would just be the same as well. So there would be no concordance with monozygotic twins. And obviously multifactorial conditions, there would be a bit of both and that sort of thing. So we go back to my busy old dad there in Colchester study in 1930s. He was looking at all these children who had mental retardation, as it was called then. And he found that children with very severe mental retardation were more likely to have severely affected SIBs than those with milder retardation. But it was also very notable that their parents were more likely to have normal intelligence, whereas the mild mental retardation were often had parents who had slightly reduced intelligence themselves. So the suggestion was that the severe cases were monogenic, caused by some single gene problem, and the other ones were polygenic. And so I think that does show the situation in many conditions where there are a small proportion which are due to single genes, but the rest is a whole mixture. So what are the non-Mendelian inheritance genes? And what are the effects? What are the causes of non-Mendelian inheritance? Well, we talked a little bit about epigenetics and imprinting, and there's triplet repeat expansion and mitochondrial inheritance, which I thought we could just talk a bit about. So epigenetics is really important. It controls gene expression, and X inactivation is an example of this. You know that in women, females, the X is inactivated. One of them is inactivated by some kind of methylation situation. But there are some epigenetic markers which are maternally or paternally imprinted. And that's rather interesting because the ones that are expressed from the paternal gene tend to be the one that encourage growth and all of that. You know, men want to be big and strong, whereas the women's one tend to be looking after the growth of the what they call the suitcase is the placenta and things like that, so to keep them all safe and sorted out. And then changes in gene regulation are implicated in some diseases, which you know about, particularly obesity and diabetes. And some imprints seem to survive from one generation to another, such that women who have been subjected to defect in diet and perhaps starvation in some cases, their offspring tend, or even their grand offspring, tend to be obese because they turn on the genes that allow them to get fatter, because that's thought to be a good idea. So all sorts of life events may affect gene regulation. How does it work? Well, it's tissue specific, as I mentioned before. Your liver is different from your brain, and so that's going to have different regulation of genes. Promoters are controlled by transcription factors, and enhancers are usually tissue specific. One of the things that is important is DNA packaging. You get chromosomes which occupy specific territories in the nucleus, but this can be relaxed or tightly packed. And if it's tightly packed, it tends to be inactive. You can't get transcription from the tightly packed DNA, whereas euchromatin can be packed and unpacked and active. There are certain genes which are expressed in a parent of origin specific manner, as I mentioned, and which are usually the growth promoter ones or so on. And also you get conditions which can be due to abnormal parent of origin imprinting, such as Angelman syndrome or Prader-Willi syndrome. And these imprints are removed and re-established in the germ cell lineages. And I don't know if you know about SDHD gene, which can give abnormalities, give you risk of susceptibility to paragangliomas. But this is only expressed if it's inherited from the father and not from the mother. So it's very important to know about that when you're counseling these families. Um, I think, yes. And I wanted to talk a bit about anticipation as well. Anticipation is the occurrence of a disease at an earlier age and in a worse phase than as the generations go on. So it's usually due to repeats, triploid repeats, such as triplet repeats, such as CGG, for instance. And these can undergo expansion during meiosis, and they can affect the protein, such as in Huntington's disease or myotonic dystrophy. And it's often seen in neurological diseases. I don't really understand why. So the disorder gets worse with ensuing generations, as with Huntington's, and also Fragile X syndrome. And in myotonic dystrophy, this seems to be worse if you inherit it from the mother. So it can get worse and earlier onset in the children of an affected mother. You can see this diagram here of a child in the middle. The little boy has got a much more severe condition than the mother, who just you couldn't see just from the photo how bad she is. And another thing, I remember this was discovered in the 1990s, and I remember being absolutely knocked off my chair when I first heard about this, about mitochondrial disorders. So the idea is that these conditions, again, usually neurological conditions, which occur due to mutations in the mitochondrial genome and not in the DNA in the nucleus. And this was very, very variable phenotype, and it was almost exclusively transmitted from the mother. And the descendants of males who were affected tend not to be at risk. And of course, the reason for that is that the sperm doesn't have much in the way of cytoplasm, and that would be where the mitochondria are. So you inherit just about all your mitochondria from your mother. So here you would have an abnormal mitochondria, a little black thing here, and these are not in the nucleus, they're in the cytoplasm. And so there'd be a sort of random effect in each egg that the mother made, such that some eggs would have a lot of copies of the little black, the abnormal DNA in the mitochondria, whereas some would have, not very many, some would have a lot. And the phenotype would only be expressed in the ones that had a large proportion of the mitochondria would have these mutations. So there's a sort of threshold, but you can see that that is completely other from Mendel's type of inheritance. And here's just a phenotype showing you there's the condition being yellow is only inherited from the mother. Now, I think I was going to do myself a favor by going into cancer genetics, because that's where I've been concentrating my efforts for the last 30 years or something. And just if you think back to the history, it's always the people who have to just be aware all the time of something unusual. So they say, treasure your exceptions. It's very important. If you see something unusual, don't just throw it away and think, oh, that's boring. It may just give you the very new idea of a completely new condition. So with Cripps, for instance, in the 1880s, he found a brother and sister who had polyposis of the rectum and colon. And this is, I think, the first description of hereditary polyposis. And then Dukes in 1930 started describing this in much more detail, and found that these people with this particular condition had multiple adenomas in their bowel. In the 1800s again, there were various people were just noting family history, families which had a large proportion of people with a particular cancer. And this poor man Broca had discovered that his wife's family had a very large proportion of people, women who'd had breast cancer, as you can see from this pedigree. And I believe that very sadly his wife did in fact die from breast cancer. And similarly, Lynch syndrome was first identified in this family, they call it family G, where there were large numbers of colon cancer and uterine cancer in this family, inherited in an autosomal dominant manner. So again, these were people describing these unusual families, and these were absolutely pivotal in finding out about the conditions. And there's my dad again looking at breast cancer cases. He found that relatives of 510 patients, he just found that there was a higher proportion of women with breast cancer in families where it had already occurred. So that, again, he was showing that there must be some kind of inheritance factor. And the point I wanted to make about this particular slide was that FAP, familial polyposis, was first sort of properly described in 1924. And it was, I believe it was started in the St. Mark's Polyposis Registry, and they started registering all these cases of polyposis and setting up a specialized center. And they started to recruit patients so that they could start looking after their relatives who they assumed might be at risk of the condition. And that really sparked the idea that perhaps if you set up a clinic where you could see patients who had these potentially heritable colon cancer susceptibility conditions, they could be ascertained and followed up, and their relatives followed up. And so that was really the start of cancer genetics clinics, certainly in Europe and America. And so the first clinics, I guess, were being set up in the 1980s, although I believe that in Canada they're a bit earlier than that. And what they were doing also, they weren't only ascertaining the people who had polyposis, but they also started looking for people who got a family history of cancer. And Joan Slack, for instance, with Richard Houlston, had worked out that if you had, say, two relatives with colon cancer, and particularly if they were young at the age of diagnosis or they'd had several cancers, the risk to your patient was higher. And the more relatives you had with this cancer, the more your risk went up. And they started offering them colonoscopies. And this was quite unusual and revolutionary at the time. And the Lynch family, I believe, were doing exactly the same in America. So then you go back, say, okay, there are genes involved. So what are the genes? Well, I think you all know about Knudson's two-hit hypothesis. He came in with this in 1971, where he found that bilateral cases of retinoblastoma, who he assumed had the genetic form of the condition, seemed to have a very straightforward age at diagnosis related to percentage of cases. Whereas the unilateral ones, which were thought to be sporadic, had a rather different curve, which suggested that there were two hits, as he called it, which were necessary to initiate the cancer. So that got caught on as the two-hit hypothesis and led to the development of the idea of tumor suppressor genes, which inhibit cell proliferation and control apoptosis. So they're sort of gatekeepers. And it's interesting that you can, of course, get conditions which are inherited due to faulty tumor suppressor genes, but they mostly do have phenotypic abnormalities, which I'm sure you're all aware of, apart from the actual cancers themselves. And one of the important things is that you often see lots of heterozygosity in the actual cancer, because as you know, if you've got a mutation in the second, I don't know if I can point to this, can I? Oh, yes, I can. So if you haven't got the gene, then you've got to get a mutation in one of the copies, and you need another mutation or a deletion in the second copy, which you would find in the tumor. But if you've got the inherited form, then you have the mutation there already inherited. And so all you need is one hit to get the second, to lose the second tumor suppressor gene. And the detection of this of the where the deletion is a clue to where the tumor suppressor gene is. And of course, that was very helpful in identifying some of these genes. And so it was very particularly interesting from my point of view, because I remember sitting in a clinic with Vicky Murday came rushing in to talk to Walter Bodmer, who was one of the king people in cancer genetics, saying, "Oh, I've found a patient who's got polyposis and who's got a deletion in chromosome 5, and in chromosome 5, and that must be where the polyposis gene is." And so they broke open a bottle of champagne, I believe, straight away. And indeed, it was in that region. In fact, poor old Bodmer wasn't the first person to find the gene, but he was very nearly the first. But it showed you that finding the deletion in that gene in the chromosome gave you a positional location for it. The same time as all that was going on, people started doing linkage analysis in big families. Now, the problem with identifying genetic susceptibility genes is with the high penetrance risk alleles, it's relatively simple to do linkage analysis and positional cloning, because if people who've got the gene tend always, or very high chance that they will have the disorder. So you can do linkage analysis, but you need big families. So this is the sort of low-hanging fruit, the penetrance genes. So I think I'll skip that one. So they started developing these linkage consortiums to get enough families where you could do proper studies. And the Breast Cancer Linkage Consortium was set up in 1989 just for that very reason, and it allowed the identification of BRCA1 and BRCA2. Mike Stratton was involved with BRCA2, I think it was. And I was at a meeting with him once, and they thought there must be a BRCA3 because there was still quite a lot of cases they hadn't explained. And we were in a harbor, and he saw there was a boat out there which was called BRCA3. So he rushed off and bought a camera and hired a boat and went out and took a photograph of this boat. But in fact, BRCA3 was never really identified. The other heritability was due
I'm
to.
curious to know there's a boat called BRCA3.
I know.
So
He
did you get
didn't.
the picture?
No, he did. I never got the picture. That's a shame,
Oh,
isn't it?
yeah.
You can write to Mike Stratton.
Okay, I will write to Mike.
Yeah. Anyway, and the other interesting thing was that with the big families for colon cancer, they set up these big groups looking for colon cancer genes, and they found that the tumors in Lynch syndrome had this mismatch repair defect, and so they were looking for mismatch repair genes which were identified in yeast, which are homologous to MLH1, MSH2, which obviously gave a clue to the possibility that those genes might be the ones that are faulty in Lynch syndrome in people. And indeed, that was found to be the case. The other thing which was nice, which actually was much later, was the discovery of the gene causing some forms of recessive polyposis, which was the MUT-YH gene. And they found that because they had this family with an autosomal recessive form of polyposis, as you see there, where the adenomas were found to have an excess of particular kind of DNA transversions, which was characteristic of DNA damage due to oxidative damage to guanine residues. And so they thought, aha, this was people in Cardiff. They thought, let's have a look at the genes which repair oxidative DNA damage. And I think there are only about 4 of them. So they started with one, and lo and behold, that was MUT-YH. So I must say, it makes you very jealous of people who do that. But it was a very nice demonstration of thinking about the possible cause of a condition and going back to actually finding the gene that causes it. But it's a recessive condition, so it's differently inherited from polyposis, but very clinically very like. So what else can give you cancer susceptibility? Well, I think as an overall, obviously this is very simplistic, but we've talked about tumor suppressors and DNA repair genes, but oncogenes and also VEGF and so on can also be involved with this. So there are a few conditions which are due to inherited oncogenes which are sort of turned on, but as you can imagine, it's a bit difficult to see how you can have a turned on oncogene and survive, really. There are a number of childhood syndromes, but this is the RET oncogene, which is involved with MEN2, which, as you know, causes medullary thyroid cancer and pheochromocytoma and so on. And there are some childhood conditions, Costello and Noonan and cardiofacial cutaneous syndrome, which are mostly defined by the developmental delay and short stature and those sorts of things, but they do have an increased risk of cancers. So it's important to know about that and follow up these children carefully. Then the angiogenesis pathway, you probably know about von Hippel-Lindau disease and SCHB and D and so on, which are causes of these conditions, which causes increased vascular activity and vascular growth, and things like these very, very vascular tumors, such as hemangiomas and hemangioblastomas, as in von Hippel-Lindau. And basically, it's a terrible slide, it turns things on. Basically, normally the von Hippel-Lindau disease gene sticks onto one subunit of the HIF gene, so it stops it being it stops it working and allows it to be degraded. But if there's something wrong with the von Hippel-Lindau disease gene, as here, oh dear, as here, then it allows the two subunits of HIF to stick together, and then it works, and it goes down and stimulates VEGF and TGF alpha and so on. Those promote growth of blood vessels, which then promote these tumors, which is rather similar to the effect of hypoxia. And so that's a rather interesting, different way of causing a susceptibility to particular cancers. So I thought I'd just whiz through this, but I don't think there's a great deal of time left. And since you are clinicians, you probably know all about these, but I'll just go very quickly through these. Just thinking about colon cancer susceptibility, there are
So,
polyposis. You
Shirley,
think perhaps I should, yeah.
you can go with the description, and these are not clinicians. So these are people from genetic counseling background and others life sciences background.
Okay. So, I mean, I can talk very
You can
briefly
take your
about,
time, whatever time you want to take. It's a
okay.
pleasure to listen to you.
All right. Good.
Okay, good.
Well, okay, we'll talk
And you have
about,
a bright sunshine in London.
I know it's good, isn't it? Very
Yeah,
unusual.
so very unusual.
You just had a big storm.
Yeah, yeah, you had a great storm. I mean, a storm, I should say great storm.
Well, we've had, anyway, yeah, last weekend was pretty
Yeah,
miserable.
yeah.
Anyway, okay, so polyposis syndromes, well, they're mostly conditions where you get obviously polyps in the colon, and they have different histological types. And I just wanted to mention as we go along that it's very important what the pathologists do and know about polyps, because they can tell us a lot about the sort of condition that they're dealing with. So a lot of colon cancer syndromes are caused by faulty gatekeepers. And as I mentioned, they quite often do have quite a lot of phenotypic abnormalities as well. And this is just a selection. So Gardner syndrome, I'm sure you all know, they have thousands of colonic polyps. They begin from about the age of 7, and without colectomy, you can see you can't leave somebody with a bowel full of polyps like this, because one at least or several will develop into colon cancer eventually. So there are a lot of precursors to cancer which develop in these conditions, but they are very sluggish. Most of them don't turn into cancer. So that's one of the characteristics here, where they grow very slowly, but there are a heck of a lot of them. And the penetrance is about 100% for these tumors to develop. And the risk of extracolonic tumors is also there. But they can have these other features like osteomas and epidermoid cysts, and these things called chirpy at the back of your eye, these dark patches in the back of your eyes. In fact, I've got one, I'm told, but I don't think I've got polyposis. Anyhow, and then you see they get lots of polyps at a very young age, which is compared to the adult normal population, very, very much earlier age, and many more profuse polyps. And the gene which causes that is a tumor suppressor, and it acts very early on in the development of adenomas. So it's one of the very first abnormalities which allows adenomas to develop. So that's, I guess, the reason why they get lots of these precursors, but not necessarily cancer initially. And when the gene was identified, it became very interesting that different mutations in the gene could cause different degrees of polyposis and different other things as well. For instance, you can see that classical bad polyposis is caused by mutations in this region, whereas very severe, I remember everyone used to say, oh, the 1309 mutation was a really bad one and a very common one as well in polyposis. You only get thyroid cancer, it seems, if you've got a mutation right there. Now, why? I have no idea. And you can get these black dots at the backs of your eyes with mutations in this region there. Ooh, help. Oh God, gone back. Back. Where I go? Oh yes, here we are. Oh, shut up. Sorry. But you can get very mild degree of disease if you get a mutation at this end or indeed at this end of the gene. So we need to learn a lot about the different functions of the different parts of the gene. But it is very interesting. Then just to mention Cowden syndrome, it's a completely different condition, much rarer than polyposis. But again, they can have multiple hamartomas. Now, it's a different kind of polyp that they get. These are not adenomas. They are quite distinct and slower growing, so the risk of cancer is lower. But you can see that they got all sorts of other problems as well. They can have macrocephaly, they can have melanoma, they can have endometrial cancer, and very lumpy breasts and an increased risk of breast cancer as well. So it's a very variable condition. Juvenile polyposis, again, a different kind of polyp. It's also not an adenoma. And again, they can have other physical abnormalities. Again, it's a tumor suppressor gene. There's one of the polyps you can see. And the genes causing this were probably identified in the late 90s, SMAD4 and BMPR1A. And some of these people have hereditary hemangiectasia, telangiectasia, because these genes also affect blood vessels and occasionally other similar kinds of genes can give rise to this kind of polyposis. So that's another tumor suppressor gene situation. And Peutz-Jeghers syndrome, another condition I just thought I'd mention because, again, there are lots of different physical abnormalities in this. They have hamartomas, but they have these very striking freckles on the face like that. But they have, gosh, I don't know what that is. Sorry. They have a risk of bowel cancer because they have these very specific polyps. But there's a picture here on the left-hand side. The sort of polyp you get in Peutz-Jeghers syndrome is not a great number like you see in familial polyposis, which is the picture up above. But here's one. And if you look at that under a microscope, it's very, very specific for that particular condition. Whereas that's an adenoma, you see, that's very characteristic. But this is very different. It has these sort of bits of muscle fiber going up and through it. And it's much less likely to become cancerous than an adenoma. But the point of that is that the pathologists are the ones who tend to recognize this, and they often refer the patients because of the histological type of the polyp. So that's rather an important thing. But the gene that causes Peutz-Jeghers syndrome is also a tumor suppressor gene. And then the last ones of this I wanted to mention is a very rare condition called hereditary mixed polyposis syndrome, which is due to a mutation in GREM1. And I only mention this because we identified it in a very large Jewish family in North London, and they knew we were very interested in them, so they had a party, and myself and another colleague went to this party, except that we went upstairs and took all the blood samples, and they went to the party and had a great time, and we didn't get any of the party. But anyway, we got the mutation, which is much more important. And it's a common Ashkenazi duplication, which was identified in about 2012. And it's also a tumor suppressor gene. Okay, back to caretakers. And these are genes which prevent DNA damage. And when they're faulty, they allow mutations to accumulate. So the cancers develop rapidly once the precursor is developed, particularly, for instance, in Lynch syndrome, which you know about. We'll talk about that in a mo. But conditions which are very well known as caused by DNA repair defects are Lynch syndrome, mute YH-associated polyposis, and of course, breast cancer. And if you see, there is a big interaction between the DNA repair genes, and so you get the Fanconi genes, the breast cancer genes, and then ataxia genes, and then the MMR genes causing Lynch syndrome, and the base excision repair genes as well. And they're all part of the same sort of pathway. There's a lot of interlinking between them. So just talking a little bit about breast cancer, of course, you know it's very common. BRCA3 was never identified, really. The second lot of genes which cause breast cancer are usually moderate risk genes, such as CHEK2, ATM, and so on. And mutations in the Fanconi anemia genes and so on. And you can see again this diagram of the Fanconi anemia genes that are involved with that. Now, what became clear is that, as I said, there are these high penetrance genes here, which can cause breast cancer. And you can see that in Cowden, of course, there's an increased risk of breast cancer as well. These are the moderate risk genes, and I'm going to talk about moderate risk genes shortly. And then these are the SNPs, these are the variants, which are very common, each of which has a very mild effect, sort of 1.2 at most relative risk, whereas these will have a relative risk perhaps of 1.5 to 3 or something. These can give you a very high relative risk. So that's a very important theme to remember. And you see the same sort of thing with ovarian cancer and, of course, with colon cancer as well. The SNPs here and the moderate risk genes here, and then the very high risk genes, which thankfully are very rare. So jumping back quickly to Lynch syndrome, here's old Henry Lynch. He and his son, Patrick, were the ones who identified these big families in America. And they were very interested in this family because at the time, polyposis was the only known hereditary disorder causing colon cancer susceptibility. But they found that these people didn't have polyposis. So it was sort of non-polyposis condition. And they defined hereditary non-polyposis colon cancer and started screening people. Now, remember, Lynch did actually say he'd been told by his boss he must stop screening these guys and get on with his real job. But it was a good thing they didn't listen to his boss. Anyway, and so as you know, it's caused
Sorry,
by...
what was his real job? What was his
he was an
background?
ordinary gastroenterologist, sort of doing ordinary
Okay, he was an
gastroenterology.
ordinary
Yeah.
gastroenterologist.
No, he told me
Okay.
that, and I thought it was quite amusing.
Okay,
Anyway,
so
don't
he,
listen to your boss is one of the
yes.
take-home messages here.
We all should not listen this.
Um, okay, so anyway, you know that microsatellite instability is one of the hallmarks, and it's due to a failure of mending a particular kind of DNA defect. And most Lynch syndromes are caused by mutations in MLH1, MSH2, 6, and PMS2, but there are these unusual cases that can be caused by methylation of the gene, or this fascinating one caused by EpCAM mutation, which causes hypermethylation of the MSH2 promoter. But thankfully, those ones are rare. And this is just a reminder again of the importance of the pathologist. So this is staining for MLH1 in a tumor, staying in beautifully nicely, staining up. And then again, staining for PMS2, which sticks to MSH1 in the tumor. Whereas you can see MSH2 is absolutely not staining up in the tumor at all, and MSH6 isn't staining up. And again, they form a complementary, they stick together. And so that's the way the pathologist can recognize that it's likely to be due to MSH2 mutation causing Lynch syndrome. I'm not going to go into all of this. You know, there are very high risks of other, particularly uterine cancer and sadly ovarian cancer in Lynch syndrome. I won't stick to go on to that, but the very characteristics of the cancers in Lynch syndrome are that they occur at young age of diagnosis. They're poorly differentiated. And if you look at them, they have a high Crohn's-like lymphoid infiltrate. The interesting thing about that is it suggests that there's a high immune infiltration. So the host, the person with Lynch syndrome, is actually attacking their tumor more than you would expect them to do in an ordinary person without Lynch syndrome. And so that gives a clue as to the sorts of causes of this response. And we'll come back to that in a minute. And they often do survive better from cancer, although not always. I remember having a couple of patients who'd had about 4 or 5 different cancers and survived them with Lynch syndrome. So I think part of this is the immune response. People who've got two copies who've got homozygous or biallelic germline mutations in the MMR genes have a very high risk of cancer. But interesting thing about that is these are children, they get these cafe au lait patches, which is just interesting, and I don't yet know why. But what you seem to find is that the parents don't have a family history of Lynch syndrome because the genes that give rise to this autosomal recessive childhood cancer susceptibility are usually the milder genes, like PMS2. So you can see here, that PMS2 is much more likely to cause this recessive condition than in the adult form because it's a much milder gene, if you like. Whereas the strongly predisposing genes probably don't allow for survival in children who've got biallelic mutations. And they get different sort of severity, different types of cancers, but it's a nasty condition. Then just to skip through, there are a few adenomatous conditions which are caused by DNA repair defects like poly and so on, polyposis, and MSH3 polyposis. I think I'll skip those. But so the key points with polyposis is that mostly they're due to these tumor suppressor genes, but they can also be due to DNA repair defects, and they have a rather different clinical phenotype. So going back to finding genes, so we found an awful lot of the high penetrance genes. So we're thinking about the intermediate ones. Well, you often have to find those by thinking what they might be and looking for them in a rather more intellectual way. For instance, things like CHEK2 were identified because people thought, CHEK2 has a similar kind of function to BRCA1 and BRCA2, so maybe that one also predisposes to breast cancer. And they found that in fact it did, and it also does predispose to some other cancers, but it was identified because of its function. And you can't usually find them through linkage because, as I said, it's reduced penetrance. So not everybody in a family would actually have the disease. And also people without the allele may still get the disease with a phenocopy. And the other problem with the moderate risk genes is that it's quite often difficult to know how pathogenic they are. And effective management guidelines may be difficult and may not be clear. And cascade testing doesn't mean that unaffected family members can be neatly categorized into high or low risk categories. So they're much more difficult to use for genetic counseling, particularly if you find them in an unselected case where there isn't a family history of the cancer. And just to think about that in a bit more detail, because a lot of people now are doing genome-wide sequencing, and this is a big thing which is coming in now, where people are going to be told their genomes, and they're going to come to shaking their copy of their results at you. And one of the big problems is that you find a mutation, for instance, in CDH1, which causes a very high risk of gastric cancer in familial cases. In a sporadic case, it may not cause a very significant risk of gastric cancer at all. And in fact, there are some studies which have shown with the different genes that if you find a gene mutation in a potentially quite high penetrance gene in a non-selected case in a population series, that it may have a much, much lower penetrance. And it's really hard to know then what to do, whether when you find it in an unselected case. Do you take their stomach out or not? Big, big important thing. Okay, and then the last bit of all of this is these common variants. Now, as you know, there's an awful lot of talk about common variants now, and what do we do with them? And obviously, you can only find them by doing large numbers of case control studies. And you screen these en masse using gene chip technology. And we find that these single nucleotide polymorphisms that confer an increase or decreased risk by association. And I do remember that when this first came out, I had a chap who turned up in the clinic and he said, oh, he had 3 high-risk genes for colon cancer, and he was going to die and everything. And I had to explain, look, these were really, really small increases in risk due to those 3 genes. He may well have 5 other genes which are reducing his risk, but he doesn't know about them, and all sorts of other things are going to affect his risk. So it's sometimes really hard to explain to people what the significance of this is. You have to be very careful. And the variants usually themselves have a relative risk causation of less than 1.2, and several hundred loci are involved. So it's very important to get this right. And they only account probably for 6% of any excess risk at all. And you could see from the top of this slide, of course, it's only a proportion of stomach cancers and breast cancers which are due to strong genetic factors. This was just a very old slide. I made it in 2003 or something like that, where they were just beginning to find the genes, some of the first SNPs that they found that were associated with colon cancer susceptibility. And you can say, just interestingly, they are genes, some of them, which you might imagine had something to do with colon cancer susceptibility, like the TGF-beta pathway or SMAD7. Those are genes that are already potentially likely to be associated. But then they went on and found a lot of other genes which you just never heard of. So people started using these polygenic risk scores, and Boadicea, which you may know about, is one particular one developed in Cambridge, which in incorporates family history, mammographic density, life behavior, and so on. And it's very difficult to do as a clinician. You sit there and think, oh, how fat are they? How many children have they had? Did they breastfeed? All these things you've got to put into this PRS. And it comes out with a score of what your risk is, which of course is a bit scary because it might be wrong. Or it might be somewhat wrong. And in England, they're starting to say, oh, you've got to have a PRS score of above so-and-so in order to be allowed to have MRI scans of the breasts, for instance. So you have to be very careful with these. And what was I going to say about those? And so what we're doing with this is we're looking at the sort of genome, the whole genome, the effects of all the genes in a person, and also their lifestyle and so on. The other important thing about polygenic risk scores is that they've been done mostly in white, Caucasian, European chaps. And so you may not be the same if you're looking at a completely different ethnic group. So you have to be, again, very careful about deriving risks in people from different ethnic groups. So then to go back to your genomic risk, wait a minute, did I say about this? So if you now have somebody coming to you and they say, I've got my genomic information in this box here, and can you tell me whether it's any good? Well, as I said, some variants may only predict disease if there's a family history of that disease. If it's a sporadic situation and there's nothing in me or my family which would suggest I'm at risk of that cancer, then maybe the risk isn't too bad and not worth doing anything about. The other thing, of course, is you've got to be a bit careful if you don't suddenly find out that your father isn't your father, which, of course, is not a new problem in genetics. And also, you may have to start, if you do find something which you think is very important and relevant, you may then need to talk to the patient about contacting their relatives to see whether they want to be tested for the same condition. So it's very complicated and difficult sometimes. I think we've talked about that. So we haven't got really much time. I think, shall I leave out the tumors because you've had enough of me? Oh, Rajiv's gone.
Yeah, no, I have not gone. I think at tumor part we can leave it.
I think so.
Yes,
Okay.
we can
I'll
finish
just
it.
go to the last slide. Where are we? Oh, no, actually, I'll tell you one thing I'll just tell you about before we go is targeted therapy, because you know that you can use anti-VEGF treatment for people who've got von Hippel-Lindau disease. And because you know that the condition is due to that sort of problem. Sorry, I won't want to do this. Chemotherapy, Lynch syndrome. That was the only other thing I wanted to say, because we were saying that in Lynch syndrome, the tumors look as though they're very antigenic, if you like. And that is absolutely what is the case, because the tumors have got an awful lot of DNA mismatch not repaired. They've got a lot of abnormalities due to the failure of the DNA repair in genes such as APC and so on. And not only are they very active, but they also are new proteins which the body hasn't seen before. And if there are an awful lot of these neoantigens, then you can raise an antibody response to them. And I think that is why they seem to raise much more of an antibody response than other people who don't have Lynch syndrome. That was all really. Oh gosh, finish. There. Thank you very much. I'm sorry I went far too fast and made too many jokes.
Thank you so much. That was really great talk. I wish we could have had more time, actually.
Yeah, Shirley, it was really very nice. Hello. It was wonderful talk, Shirley.
Oh, well,
And
thank
I
you.
don't think I'll allow question and answers now, but they will now. We have your email. All our
Okay.
students will write to you, And
Okay.
I'm sure they were asking me, can we ask her for more lectures? I said, I'll ask her.
Oh, right. Well, it's
And
a
they liked
great
your
pleasure.
style.
It's a
They liked
great
your
pleasure.
style of teaching.
Thank you. Well, it's a great pleasure, and I love being still involved with you guys. It's very important. I think, especially in these awful years at the moment, to have
Mm,
people collaborating,
mm,
and
mm.
being friends.
And Shirley, this February, our meeting is on genes less known and VUS. Like you talked about the genes intermediate penetrance and low penetrance genes. So
Yeah.
2 days we'll be talking only about these genes. So it
Oh
is in
God.
February third week. So would
Okay.
it be possible for you to come?
To come.
Yeah,
Well,
you
I'll
can
have
think
to
about
think
it.
about that. I'll have to think about that. Trouble is,
Yeah.
it's my birthday, and I won't tell you which birthday it is, but it's
When is that? We'll have a grand celebration. When is that? When is your birthday?
22nd February.
Oh, 22nd February, 22. I am 2nd February.
Oh, really?
Yeah,
Ah, nearly.
uh, yeah, yeah,
Well, let me think about that,
yeah,
but I'd
yeah,
be very pleased to be involved
yeah,
anyhow.
yeah.
Where? In
Okay, thank
Mumbai?
you. Yeah, in Mumbai, yeah. Same.
Okay. Fine.
Okay,
Okay. Thank you very
bye,
much for
bye. So
listening.
thank you
Okay.
very much. Thank you for, doing so. We will also end the meeting. One second.
Okay.
Can you stop sharing the slide? Yeah, I can.
Yeah, I can, I think. Anyway.